Greetings, my name is David Carmel, and I'm Senior Vice President of Public Affairs and Communications for eGenesis, a regenerative medicine company using breakthrough gene editing technologies for the development of safe and effective human-compatible organs, tissues, and cells. We are proud members of the Alliance for Regenerative Medicine. Personally, I've been in the field of regenerative medicine for 15 years, since I worked on the ballot initiative that became the California Institute for Regenerative Medicine. I'm happy to present on the company's behalf for our first meeting on the Mesa. eGenesis is revolutionizing the field of transplantation thanks to our world-class multiplex gene editing platform that was spun out of Dr. George Church's lab at Harvard to address the historical challenges for xenotransplantation. We have a bold vision to create an unlimited supply of human-compatible organs, tissues, and cells. We have already demonstrated the ability to produce durable xeno organs with a near-term pathway to IND for our lead programs in kidney and islet cell transplantation. In the US today, more than 20 people die every day due to a lack of available organs for transplant. There are 110,000 people on the transplant waitlist in the US alone. Initially, we want to provide a new therapeutic option to these patients using immunosuppression, which is the current standard of care. Secondarily, we expect to be able to reduce or eliminate immunosuppression, which would lower the incidence of infection and cancer for transplant recipients. Eventually, we want to revolutionize the practice of transplantation from surgeries that occur at any time of day or night with enormous cost to the medical system to elective procedures for patients who don't wait years and don't want to suppress their immune systems. Our broad pipeline spans organs, tissues, and cell therapy. Our lead programs are kidney and islet cell transplantation. Kidney is an optimal initial application given the high unmet need, technical probability of success, and the ability to transition a patient to dialysis in the event of a graft failure. Our islet cell program is addressing the type 1 diabetes market and could eliminate the need for insulin. We have also done liver perfusion, heart, and liver transplants into non-human primates. In the short time since our founding, we have significantly de-risked the field of xenotransplantation. Our foundation is the intellectual property out of Dr. Church's lab, and we raised our Series A in 2017. In 2019, we showed 9-month NHP survival, that is non-human primate survival, with a xeno kidney and closed $100 million Series B financing. This year, we acquired our production partner, IC Biotech, expanded our collaboration with Massachusetts General Hospital, and demonstrated consistent non-human primate survival, including a non-human primate who is currently nearly 10 months post-transplant and doing well with normal kidney function. We expect steady and meaningful news flow in the next 24 months, including in vivo expression of our transgenes and survival data for our lead programs. For xenokidney, the FDA would like to see 6 to 12 month survival for 6 non-human primates before approving an IND for the first in-human study. We presented data at the end of last year, which showed that a non-human primate lived more than 9 months and have repeated that again this year. We also expect to begin first in human studies for both xeno and islet cell transplantation in early 2023. We have built a highly experienced management team, including the most recent additions of Peter Hansen as COO and Mike Curtis as president of R&D. Dr. Church, co-founder of the company, remains involved as a member of our scientific advisory board. Dr. James Markman, Chief of Transplant Surgery at MGH, sits on both our clinical and scientific advisory boards and is very active with the company. We expect to continue to add highly skilled and experienced people as we grow the business and advance our assets into human studies. The field of xenotransplantation is not new. Cross-species transplants have been contemplated for over 100 years, predating allotransplantation. The pig is the optimal species for humans due to similarity in organ size and homology and relative ease of breeding. Two key hurdles have prevented xenotransplantation from advancing. The first is the risk of transmission of porcine endogenous retroviruses, or PERV, which are embedded into the pig genome. Significant resources went into xenotransplantation in the 1990s, but the inability to eliminate PERV hampered the field. The second hurdle is overcoming the immune response to molecular incompatibilities between donor and recipient. In 2017, we published a seminal paper in Science showing inactivation of all known PERV sequences in the porcine genome. Those genetically modified PERV-free pigs are fertile and the edits are heritable. We have produced multiple generations of PERV-free pigs and have seen no evidence of vertical transmission from sow to piglets. Having successfully overcome the virology hurdle, we are now focused on addressing the immunology hurdle. We take a comprehensive approach to addressing the molecular incompatibilities that drive all stages of immune rejection. In hours after transplant, hyperacute rejection occurs. We modulate antibody binding and complement factors. In the days after transplant, we modulate coagulation to avoid vascular injury. Over the longer term, cell-mediated and chronic rejection can occur. We target NK cells, macrophages, to manage the antibody response. eGenesis builds on decades of science, which has identified gene targets across the immune rejection process. Leading academic institutions have shown by targeting specific genes, xenotransplantation can have positive results with non-human primates living for months with various organ types. We are the only ones targeting all aspects of rejection, which we think is critical for long-term survival. We have an end-to-end production process that takes place in 3 different locations. In our laboratory in Cambridge, Massachusetts, we do all of our gene editing of a porcine fibroblast. That cell is then cloned to make a colony so that we can do QA and QC testing to ensure successful edits of all targeted genes with no off-target edits. Those cells are then shipped to our transgenic animal production facility, where the nucleus of the edited cell is transferred into a pig oocyte in a process like the process used to create Dolly the sheep. The embryo is then transferred into a surrogate sow and matured after birth. The organs are then recovered and available for transplant. We are currently working with Massachusetts General Hospital to do the xenotransplants, but anticipate announcing other partnerships with leading transplant programs soon. We've made significant progress in our production and preclinical development. We've engineered many genetic constructs and have over 350 genetically modified pigs on the ground. We have completed more than 30 non-human primate transplants for kidney, heart, and liver with encouraging early results. As mentioned, we now have 2 xeno kidney transplant recipients that have survived over 9 months. The FDA has been very collaborative given the novelty of this field. And in xenotransplant, for kidney, they want to see 6 non-human primates live for between 6 and 12 months. The FDA also gave guidance that eliminating PERV reduces their concerns on viral transmission. We're pioneering a transgenic production process to supply unlimited GMP grade organs, tissues, and cells. Once we have the pigs produced with the genetic changes we want, we can create a breeding program in what's called a designated pathogen-free facility. This facility ensures that animals are regularly tested for any possible pathogens. And this facility will support pharmaceutical grade production. Our lead programs in kidney and islet cell transplantation address unmet medical needs for large numbers of patients and are rapidly advancing towards the clinic. The burden of kidney disease in the United States is overwhelming, with 15% of the US adult population, or 37 million people affected. Of the 500,000 patients with end-stage renal disease requiring dialysis, about 95,000 are on the wait list. Roughly 7,000 patients die each year waiting, or their health status worsens to the point that they can no longer stay on the wait list. The current standard for care for renal failure is expensive and ineffective. In the United States, we spend $60 billion each year with an annual cost of $100,000 per patient to treat end-stage renal disease. Only half of dialysis patients can expect to live more than 5 years, versus 2 out of 3 patients with common cancers. We desperately need new treatment options for these patients. Our first-in-human study will focus on the patient population that has the most difficulty finding an appropriate match. More than a quarter of patients must wait more than 4 years for a transplant, and each year, 1 out of 5 patients on the wait list die. Those with a high CPRA, which estimates the percentage of donors with whom a recipient would be incompatible, would be ideal candidates. Our islet cell program will initially target patients with severe type 1 diabetes, or T1D. 1/4 of the 1.25 million people in the United States suffers from impaired awareness of hypoglycemia. 20,000 of those T1D patients experience frequent and severe hypoglycemic events. Islet cell transplant offers a less invasive alternative to whole pancreas transplant, which has a relatively low success rate. We are committed to building a world-class gene editing company. We have an exclusive license to intellectual property, which came out of Dr. George Church's lab for using multiplex gene editing for xenotransplantation, as well as the methods for addressing PERV-free organs, tissues, and cells. As we have gained experience, we have patents pending for more advanced methods for producing pigs with both immunological and virological edits. Eventually, we hope to apply for a composition of matter for our source animals. Our competitive set is limited, with just 2 companies in the US who are working on xenotransplantation. Revivicor had originally tried to work on lungs, but has since moved into kidney. They are addressing some aspects of the immunology hurdle, but do not inactivate PERV. They are also part of a larger company. Xenotherapeutics is in the clinic for skin, but they don't modulate the immune system or inactivate PERV, given the nature of the temporary skin allograft. They have received limited funding to date. Increasingly, the field is moving towards the belief that it is when and not if xenotransplantation will be a reality for patients. Given the significant progress we have made, we are raising additional capital to accelerate our programs into the clinic. We plan to realize our vision that no patient will die waiting for a transplant. Thank you to ARM for putting on this conference, and thanks to all of you for joining this presentation.